Hi everyone, my name is May, and I just wanted to give a quick thank you to everyone at the event today and being so understanding that I was unfortunately not able to make it there physically, but I'm still very excited to present. My Inspired Discoveries project is based off of an internship that I did at a neurobiology lab focused on investigating the neurobiological mechanisms that mediate maladaptive behaviors that stem from drug use by examining how drugs alter things like neural activity, gene expression, and circuit function. Addiction or substance use disorder is widely recognized as a chronic, relapsing disorder characterized by maladaptive decision making, impaired motivation, and persistent drug seeking. These behavioral patterns reflect disruptions in cognitive control, motivation, and reward processing, all things that I was particularly interested in regard to my psychology background. Addiction, from a psychological perspective, is understood as a substance abuse disorder characterized by a general inability to control the use of a substance. When drug use becomes a substance abuse disorder, consequences can be seen in all aspects of life, including family relationships, financial status, and health. Over time, continued drug use leads to brain adaptations and a reduction in dopamine receptors and release, which leads to tolerance. This change can be observed in certain brain regions, such as the ventral tegmental area, the brain region where dopamine neurons are expressed, which promotes reinforcement and motivated behavior. The prefrontal cortex is also needed to regulate executive functions, such as decision-making, impulse control, and motivation. The nucleus accumbens releases dopamine into the brain, playing a significant role in processing reward, motivation, and pleasure. The dorsal striatum is responsible for creating habits and drug-associated cues that lead to compulsive drug-seeking behaviors. And finally, the ventral hippocampus associates drug use with memories such as specific environments or contexts, which makes it particularly important for facilitating drug-seeking behaviors and initiating relapse. To study these changes, scientists often use animal models like mice, which is what I used in my lab, and while mice are not identical to humans, they share many of the same brain pathways involved in addiction, allowing researchers to study both behavior and underlying biological processes in controlled ways. All experiments and procedures are approved by the Institutional Animal Care and Use Committee and are performed with the highest standards of ethics. We take great care in handling the animals, and we thank them for their contribution to science and research. My work specifically focused on understanding how cocaine affects the brain across multiple levels. Specifically, I looked at both behavioral changes like reward preference and cellular changes such as mitochondrial function. One of the primary experiments that I worked on was conditioned place preference, or CPP. CPP is a behavioral paradigm used to measure the rewarding effects of a stimulus by repeatedly pairing it with a specific environment. It's widely used in addiction research because it provides a clear behavioral measure of how environmental cues gain motivational significance through conditioning. This process parallels how drug-associated contexts can trigger craving and relapse in humans. This experiment took place over 4 days. On the first day, each mouse was allowed to freely explore both chambers of the apparatus, one marked with dots and the other with stripes, to ensure that the mice had no baseline preference, and those that did were eliminated from the study. On days 2 and 3, mice were randomly assigned to one chamber where they were confined after receiving an injection of either cocaine or saline, creating an association between the drug and that specific environment. On the final day, the mice were again given free access to both chambers, and their time spent in each side was recorded to determine whether they developed a preference for the cocaine-paired environment. For this experiment, we used a DRP1, or a dynamine-1-like protein, a virus to knock down gene expression in order to examine how drug use affects mitochondrial fission. Ideally, we would find that the DRP1 expression was significantly lower than the scramble MR, or our control group, but that's not what we found, as you can see by the graph. The results actually indicate that something interfered with the inspected gene suppression. One possible explanation that we looked at is that the viral vector used to knock down the DRP1 had reduced effectiveness, potentially due to age or just general degradation. But this just goes to show how important and sensitive gene manipulation experiments are to factors like reagent quality, and it gives us something to look for in the future. In the CPP paradigm, we also assessed locomotor activity as an index of behavioral stimulation. Cocaine-treated animals showed a significant preference for the drug-paired environment and increased locomotor activity compared to the saline-treated controls. The next experiment that took place was self-administration. Self-administration research is, again, a behavioral paradigm used to assess voluntary drug-taking behavior by allowing an animal to perform a specific action to obtain a controlled dose of the drug. The lab that I worked at used a cocaine self-administration experiment to examine voluntary drug-taking behavior. In this experiment, each mouse was placed in the chamber with two levers, an active lever that delivered an intravenous infusion of either cocaine or saline, or an inactive lever that did not administer anything. Pressing the active lever resulted in an immediate drug infusion paired with a cue light, which served as a signal that the mouse had received the drug. Over time, mice learned that pressing the active lever produced a rewarding outcome, which led to increased responding on the active lever as opposed to the inactive lever. Self-administration experiments are especially important in addiction research as they model voluntary drug taking and allow researchers to study how drugs reinforce behavior. Furthermore, they provide insight into the motivational and learning processes that drive addiction, including habit formation, cue reactivity, and relapse. I did want to give a quick warning. I'm going to show an image of a jugular catheter surgery being performed on a mouse. This kind of gives you a better idea of some of my responsibilities in the lab and also how the cocaine is administered in self-administration experiments. If anyone does not want to see those images, I suggest looking away now. For this experiment, mice undergo a procedure where a small catheter is placed into the jugular vein, which I hope you can see in the image here. It allows substances like cocaine to be delivered directly into the bloodstream, and this method ensures precise and consistent dosing during the behavioral experiments. We hypothesized that the mice that initially received cocaine would show stronger drug-seeking behavior as measured by active lever presses compared to the mice that initially received saline, and that is actually what we found. And you can see that on the graphs here if you look at the cocaine groups in green. Another experiment that we looked at was chronic cocaine exposure. The figures shown on this slide show full change mRNA expression of mitochondrial fission and fusion genes measured in the ventral hippocampus tissue punches that we collected 24 hours after chronic cocaine or saline administration. Although you see a bunch of different genes on these graphs, we're really only focused on DRP1 as it most closely is identified with mitochondrial fission, so we pulled those and they're on the bottom right of the graph. DRP1 expression is reduced following cocaine treatment, suggesting reduced mitochondrial fission and a shift in mitochondrial dynamic. This can contribute to altered neuronal function during withdrawal in the hippocampus, and this can severely affect how the brain functions during withdrawal. In humans, addiction is a chronic brain disorder that allows for the brain's reward systems to become rewired, making it extremely difficult to stop using a substance even when there are negative consequences. Over time, the brain begins to rely on the drug to feel normal, which is why addiction isn't just about willpower, it involves real biological change. Mice are widely used in neuroscience research because their brains share many structural and chemical similarities with the human brain. In both mice and humans, addiction involves similar brain pathways, especially those related to dopamine and reward. Using mice allows researchers to control variables like gene expression and gives us the opportunity to study changes in behavior and brain cells in ways that wouldn't be possible or ethical in humans. While mice are not identical to humans, they offer critical insight into how addiction works at both the behavioral and the cellular level. These findings help guide human research, improving our understanding of addiction and supporting the development of more effective treatment options. Thank you guys so much, and if anyone has any questions, I'll be on the Zoom for the Q&A portion. Thank you.
Round of applause for May. Woo! Okay, May, are you still there? Did we make it through the museum Wi-Fi successfully?
Yes, I think so.
I'll just pin you so that we're at least doesn't matter. Questions for May. All right, here's one from Dr. Pesher in the room.
Yes, thank you. Impressive work. Just to get an idea, how long had this work been? How long did it take you to do that work?
Yeah, so the addiction research at the lab that I worked at, it's been going on for decades, honestly. And the interesting thing about addiction research and research in neuroscience in general is that it's very focused on replicating their studies and their experiments over and over and over again. So, although I only showed one example of each experiment in my presentation, these are actually experiments that I've done probably 5 or 6 times by now. I've worked at the lab for about a year. So, that's just something really important to neuroscience research is just to do it over and over again to find if your findings and if your experiments are correct and if we can learn anything else from them. So yeah, hope that answers your question.
Uh, hey, May, first off, great presentation. I really enjoyed it. One thing that regularly comes up for me when thinking about experimental paradigms like this is the question of ecological validity. So how well do these experimental settings map on to real world settings that we want to generalize? So I'm just wondering if you could speak to that issue, say perhaps the first setting where you're looking at context. What sort of real life scenarios would be relevant here?
Yeah, so I can't speak exactly to the connection that my lab and the research that we do there has to real life situations. Obviously, you can kind of assume the real world implications that this research has. But just speaking generally and from probably like a psychological perspective in this, addicts or people suffering from substance abuse disorder, they tend to um, feel the drug less when they are in the same setting. So, for example, if someone is using the drug at their house, they're going to start to gain a certain tolerance for it. And certain triggers, too, like environmental triggers, can also affect relapse and affect the addiction that one has to the drug. So, while I can't give like an exact answer to that, there's definitely real world applications that you can look into with the studies that I put on my PowerPoint and the research that we found from that. But also the lab specifically that I worked at, we do drug addiction, but we also look at social stress. So I think that speaks to just kind of how broad the research can be.
Thank you. Check. Is my microphone on now? Yes. Great. I'm going to ask a two-part question while we're setting up for the last one. So thanks, May. So part one, how's the museum?
I haven't been in it actually.
Okay. Go in there after this.
Okay.
The second part, this is one of our first ever internship related presentations. I'm just really curious, how do you feel like this has impacted what you're going to do after you leave UBalt with this degree?
Yeah, that's a really great question because it's actually something that is kind of affecting me right now. This is my last semester before I graduate, so I'm, of course, thinking about what my next steps are. Originally, I had never been one to kind of like the subjects in STEM. I usually strayed away from things like math or science subjects. And I really didn't think that I was going to like this and working with animals and neuroscience, but this internship really turned things around for me. Unfortunately, one of the main things that plague my mind when I think about going into neuroscience professionally is the lack of funding that it has, especially now in today's climate. That's one of the main things my lab is suffering from is just a lack of grants, a lack of funds. So that is something to take into consideration. But yeah, neuroscience, this internship has definitely pushed me towards wanting to pursue that professionally, academically, and it's something I'm definitely considering.
I'm going to tell Lakeisha in the Career Center right now. Okay, thank you very much for May.